All right. Well, welcome everybody to the BIDS panel hosted by the BIDS maintainers. So just to kind of give a high level structure of what we're going to of how we're going to be organizing this panel is that I'm going to start off by just doing a little roundtable of introducing all the maintainers here. And then Chris is going to take us away with describing some of the derivatives and BIDS apps within BIDS. And then we're going to go into a more open format of each maintainer is going to describe a little bit of the work that they do with trying to keep BIDS moving forward and operational and keep it in the nice form that I think you all are seeing it in. And then at that time, we want to further open it up to be the more engagement and open section of this panel, to really engage with you all, answer any type of questions you have regarding BIDS, being a BIDS maintainer, and that sort of thing. So to not further ado, I think we'll start off again with these just quick roundtable of introductions, introducing all these BIDS maintainers that we're lucky to have here today. We'll start off with Chris.
Hi, I'm Chris Markiewicz. I'm a software developer at Stanford University. I think that's the main thing you need to know.
Great, thank you. And next up, I got Remy.
Hey everyone, I'm Remy. I'm a postdoc at the Université Catholique de Louvain in Belgium. Yeah, and I mostly do fMRI stuff in general when I actually don't do BIDS stuff.
Great, thank you. Next up, I've got Stefan.
Hi everybody, I'm a third-year PhD student in cognitive neuroscience at the Max Planck Institute in Berlin. And I've been a maintainer for BIDS since 2019.
Perfect, thank you. Next up, I've got Taylor.
Hi all, I'm Taylor Salo. I'm a PhD student at Florida International University.
Thank you, and I'm Franklin. I'm a Associate Director of Strategy and Operations in the Center for Reproducible Neuroscience at Stanford University in Russ Baldrick's group. Great, so that's all the folks that we've got here on the call on BIDS maintainers. And so now I think I'm gonna kind of hand it over to Chris so he can present a little bit about what the BIDS derivatives and BIDS apps are.
Okay, let me share my screen. Okay. I'm assuming people can see this. Okay, so again, my name is Chris Markiewicz. I work for the Center for Reproducible Neuroscience at Stanford University. This talk is I'm going to be introducing the brain imaging data structure, or BIDS. You can find all these slides online, and then I put a number of links through here so that if you follow along, you can click out to any of the resources I point to. Okay, so it's going to be a fairly basic overview. We're going to talk about BIDS itself, then BIDS apps, and then BIDS derivatives. So what is BIDS? It's the brain imaging data structure, and it's a directory structure based on common practices. If you've done, if you've worked in a lab and worked with neuroimaging data and anything that isn't BIDS, it's probably still going to look fairly familiar to you, at least with MRI imaging data. So one of the things that we try to do with it is build on existing standards rather than making people develop entirely new tools in order to work with it. So specifically, NIfTY, JSON, and TSV are the main structures we use. And the goal is for human and machine legibility. On the side of creating the data structure, or creating a BIDS data set, the main resources that are going to be of interest are going to be the BIDS validator, so that you can check whether or not your data set is compliant, and the specification for finding detailed information on what to do there. And so again, this is not really, or BIDS is not so much about the data as it is about the metadata and how you organize the data. So the first place that you find metadata is going to be in the file names. So subject, session, imaging modality, et cetera. And these are generally, the goal is just to put enough into the file name so that you can have a unique name. You can quickly look through the data set and find it. So we've got some terms we use there, like suffix, which refers to this last bit after the final underscore. So T1 weighted, this is a T1 weighted image. This is a bold image. This is a diffusion weighted image. And then we also have these dash separated groups, which we call entities. So the subject entity here has a label of 1, the task entity has a label of rest. You can also have indices. So if you run multiple runs of the same task, you would say run 1, run 2, etc. Then where for image level metadata, a lot of that is going to be stored directly in the Nifty headers, but even more is in JSON sidecars. And so if you haven't run across JSON, it looks like this. It looks a bit like a Python dictionary if you've run across those. And so here's an example that might correspond to a bold JSON file. So you describe the magnetic field strength of your magnet, the manufacturer of the scanner, and then interesting parameters such as repetition time, phase encoding direction, etc. You can, for tasks, you might include task instructions. You might include relationships between files. So this field map applies to this functional image, or so on. And in addition, we have study level metadata. So the main file that's required in BIDS is the dataset description. That's where you'd have name, authors. You might put in, funding information or associated publications, things like that, that don't really apply to individual files. And you can also have participant level metadata such as age, sex, handedness, diagnosis, anything that you might somebody analyzing the data might need to know to compare one subject to another. And similarly for sessions, if you have subjects come from multiple sessions, you can encode visit level metadata, or scans.tsv. You can encode anything about the specific scan. So how do you get a BIDS data set? You could do it manually, tends to be error prone and tedious. So a number of converters have been written. Probably the most prominent one is called Hudiconv, which is for converting DICOM images to BIDS data set. So on the left here, we see a somewhat typical DICOM directory, which is pretty unreadable. And so the goal of BIDS is to take all of this data and turn it into something that's both human and machine readable. I think that's the main thing to say there. If you're planning on collecting data in the future, I want to kind of push people towards this project, which is called ReproIn. And so basically, these are rules for that you can provide to the people operating your scanner, possibly yourselves, that if they name their DICOMs in certain ways, then you can convert to BIDS with basically the push of a button, which makes things a lot easier for everybody involved. It's less error prone. So you front load a little bit of work at the scanner time, and then everybody at the scanning center benefits from that, at least insofar as converting to BIDS goes. To give you a sense of what the BIDS validator looks like, you can go online to do it, and this is an in-browser JavaScript tool. So it says upload your data, but actually it's just we send the code to your browser, and then you aren't sharing your data with anybody, so it's privacy respecting. But if you can also install it with npm to as a command line program, and then you'll see this. And so this is what it might look like for here's a data set from OpenNeuro. And these data sets are valid, so they don't show any errors, but they might have warnings saying, you probably want to have an events.tsv with this bold series. In this case, they didn't actually, because the task is watching movies. There isn't really an event there. Okay, so I guess the thing that all of this enables is that it makes it easy to programmatically work with BIDS data. And so the two most common languages in neuroimaging labs these days are probably Python and MATLAB. I don't actually know for sure. but I think that's true. And so there are libraries in Python and MATLAB. So one's PyBids, one's BIDS-MATLAB that allow you to just pass it a BIDS data set and start querying information out of it. So for example, here is what it would look like for Python is you create a BIDS layout from a particular data set, and then maybe I want to select or find all the files that have this subject, Pixar02. I want them to be bold files, so they'll have the bold suffix, and I want them to be images, so they'll have the nifty GZ extension. So I get a list of these BIDS image files, and it parsed the directory to find that. I can also find any associated metadata, such as repetition time, and I can also get back the entities that you find in here. So for instance, Pixar02, you see that entity, it's right in the file name. PyBids also includes things like func as the data type and suffix, which BIDS itself doesn't really consider entities, but from a parsing perspective, PyBids does that. I'm not really sure if BIDS MATLAB follows the same principles, but it should be similarly easy to work with. And what this really allows is it allows us to step away from telling programs, here's where I put my T1 weighted image, here's where I put my bold image. Or if you're doing, say, image co-registration, you just pass it those images individually. BIDS allows you instead to create what we call a BIDS app, that you just pass it the whole data set, and that tool knows what it's looking for in the data set and knows how to find it. And so this protocol is extremely simple, and by protocol I just mean this template for command line options. And so you would take the name of your BIDS app, you would pass it your input directory, which would be a BIDS directory. You tell it where to place any outputs that it might create, and you tell it, here we're going to use participant. This is what we call an analysis level. That means basically operate on each participant individually. You could say the group level, which would be able to take data from all of the participants. Other levels include session and run, and then it might give other options. So just to give a couple examples from the Center for Reproducible Neuroscience, we have a tool called MRIQC. And again, you see just MRIQC is the tool. You pass it the input data set. You tell it this, maybe I want to put, this is a reasonable place to put the outputs, and I want to run this at the group level. Or for fMRI prep, exact same thing. So fMRI prep, input data set, output location, participant, and then I might put in an option saying, let's just run on this subject. And so you can see that there's going to be a lot of flexibility to run any kind of application. MRIQC and fMRI prep are quite different in what they do, but they take almost identical parameters. So here on the right is a screenshot of the BIDS apps webpage where we keep track of a bunch of BIDS apps that have been published. And so just to highlight some different kinds that are possible, we've got a couple of quality control apps like MRIQC, and I think this is the quality assessment protocol. There are anatomical pipelines like FreeSurfer, functional pipelines like one built around SPM, diffusion pipelines like Nutmeg. And this gets us into a place where BIDS and BIDS apps kind of mutually reinforce each other. So if I'm a researcher and I want to run MRIQC, I convert my data set to BIDS, and now it takes me almost no work to try out any number of these other apps. Whereas I might have had to spend a while getting to learn them before somebody went and made a BIDS app for them, or I had to use a non-BIDS form of the application. And similarly, there's a benefit on the tool developer side, which is if you write your tool to accept a BIDS data set, you now have a lot of people who've already put their data in BIDS for whatever reason, and they can try your tool with almost no friction. Going to try to go through this fairly quickly, but this is outside the scope of like BIDS per se, but one of the useful things about BIDS apps is, one of the things encouraged by the paper that introduced BIDS apps was to use what they call containerization, which is basically packaging enough parts of the operating system to make sure the app can run on anybody's computer. So I might write my tool on Linux, but the person running it might use it on Windows. So if I build a container and put it on Docker Hub, then you can run it on Linux, you can run it on OS X, you can run it on Windows, and you can even convert it to Singularity and run it on high performance computing cluster, such as CBrain or a cloud environment like AWS Batch. Other things that this allows, this is more of a conceptual thing than something that's implemented in a general way, is by separating out these analysis levels, it provides natural opportunities for parallelism, such as splitting over the subject level, running an independent process for each of those, which you could easily parallelize out on a cluster, and then only bring them back together at the group level when you actually need the data from each subject. And I want to divert a little bit here, I guess divert a little farther here, to talk about reproducibility, because I think this is one of the things that BIDS apps really encourages. And it does so in the same way that any start to finish pipeline would. And so just to define a couple terms here, I like this table from the Turing way. We use reproducible to mean if you run the same analysis on the same data, you get the same result. And so these tools allow you to start from your raw data, or if you have a A to Z pipeline, that you can start from the raw data and get the exact same result every time. Or if you don't, there's something wrong, there's an issue with your pipeline. And then just to complete the table, if you run the same analysis on different data and the result still shows up, we call that a replication. If you run a different analysis looking for the same effect on the same data, that effect is robust. And then obviously if you have, the goal is in science is generalizability. So I should be able to run multiple analyses that are aimed to elicit the same effect on multiple data sets and get the same effect reliably. And so that's what we call generalizable, at least in this context. So when we use open analysis on open data, that's subject to independent reproduction. And if you accept BIDS datasets, it makes replicating results on independent data easier. And then a common interface also makes independent analyses easier to write and compare. So following this, these two rows. Okay, so I don't know if I should stop and take questions, or I can just kind of keep moving. I don't know, what do you guys think?
Yeah.
Just keep moving. Okay. Okay, so then BIDS derivatives is a related concept, which is it's just the output of a BIDS app. So that's a derivative data set. Now these can output anything really. So BIDS derivatives is trying to standardize outputs that might be usable in further analysis. By the same principle of why is it useful to have your data in BIDS and you can run it on different analyses. If you've produced an output that isn't a figure ready or a publication ready figure, there's probably more things that you want to do. And so having common ways to name these things is advantageous. So the types of derivatives will typically fall into three categories: pre-processed images or otherwise transformed data for further analysis. So this is the main thing that we focus on in BIDS derivatives. Other things that are probably going to happen eventually, but haven't seen a lot of push forward yet, are statistics of interest, such as GLM stats or fractional anisotropy for diffusion, etc. And then also figures and reports are often useful. A number of tools produce those. So what do these look like? So it's still, the goal is to be as similar to BIDS as possible, but recognizing that the space of derivatives is just massively larger than the space of raw data. So one of the in the dataset description, we can include first just a quick distinguishing dataset type that distinguishes raw and derivative datasets, and then also additional metadata explaining that allows tools to record what they've done to a tool. so they may record information about what tools they've run internally, or they may just record information about themselves, saying, I'm fMRI prep version, 1.6 or whatever, and just put that in this output. And then source data sets is intended to list references to the specific version of the data set that was analyzed, to help you keep at least a basic provenance of your derivatives. So the metadata we still see, there's still the basic metadata in the file names. So, but you may notice there are some new entities, such as space or desk. So desk is description. Suffixes, so mask is a new suffix, so it's a new type of file. Extensions, so here is a functional GIFTY file. And so that's something new that has to be introduced, because those are not in raw BIDS. Additional metadata in sidecar, pretty much all BIDS derivatives, I think all BIDS derivatives allow you to specify, just a natural language description of what you have, the specific files that produced this current file. And if you've, say, put your data into a template space, you may want to indicate a spatial reference that allows people to compare across individual subjects. And so the types of common derivatives that are currently specified include basically the pre-processed or clean data. And so the idea of this is it's essentially the same as what came in. So you started with a bold series, you still have a bold series, but now it's been aligned correctly. It's in M&I space, et cetera. You might do some filtering or cleaning on your EEG or MEG data. And so the main rule in derivatives is that you need to have something in the output file name that distinguishes it from the input. So space or description may be relevant. And when you create this metadata, it should be retained from the original unless it's invalid. So repetition time for bold pre-processing is not going to change, but if you run slice timing correction, those slice times are no longer valid. They don't apply to that time series. And specific to imaging files, you can also include resolution and density metadata. A couple new imaging data types. So we have the masks and determinate or discrete and probabilistic segmentations. I think we have one more. Nope. Okay, that was it. And for when things are atlas derived, there's additional metadata for labels and atlases. And so that's basically where the official standardized derivatives are at this point. There are a number of upcoming derivatives that people have been thinking about, shared at least progress with the community, but they have not yet been incorporated into the standard. And so we do this through a process called BEPS, BIDS extension proposals. And so one of them is structural derivatives. So things like that are those GIFTY files that I mentioned earlier are going to be related to that. Functional derivatives, that's going to include, I'm trying to remember exactly. I know it includes things like temporal ICA decompositions, temporal or spatial, etc. Affine nonlinear transforms. I'll probably just stop trying to give examples at this point because it seems my brain isn't doing it. Diffusion weighted imaging derivatives, connectivity data, electrophysiological derivatives, and PET derivatives. So those are all in progress. And so for the most part, these are different teams who get together, think through their problem, put together a proposal, and say, these are the things that we think need to be added to BIDS. It can take a bit of time, and we'll probably be talking about that in a little bit as we talk about what maintaining BIDS looks like. So that's just kind of what's going on next. And here, if you do go to this site or this slideshow, are a bunch of links to things throughout this slideshow all in one place. And thanks for your time. And again, here's the URL if you need it.